Thank you very much, Chris, for agreeing to give a Build a Cell seminar, and it's all yours.
Great, yeah, thanks. So I'm Chris Kempes. I'm a professor at the Santa Fe Institute. I come out of physics originally and then moved into combinations of physics and biology, physics and ecology. And at a high level, I'm very interested in universal laws of life. So I think it's interesting that we have really good predictive theories of, for example, physics that works anywhere in the universe, allows us to predict surprising things that we haven't seen yet, like black holes, or hadn't seen yet at the time of some of those predictions, and gives us sort of universal theory. And I think we need a similar sort of thing for life. And I think that applies to then a huge, developing such a theory really helps us get a huge range of future avenues open up to us in terms of engineering new types of life, finding new types of life through astrobiology, and also just understanding the full history and origin of life on our own planet. Great. So with that introduction, I'll talk today about some of those universal patterns, mostly through the lens of thinking about individual cells. So I think typically the way we think about biology is a picture like this, focusing on the vast diversity of organisms, both in terms of different morphologies that an organism might have, the huge range of metabolisms and physiology that these organisms possess, the environments they live in, the competitors they have, what communities they form. And often we would say, okay, the only way to understand one of these species is through understanding its very detailed evolutionary history, the niche it lives in an environment, and the set of competitors it may have, right? We have to understand everything about evolutionary dynamics. And for some questions, I think that's true, that there are certain aspects of an organism that you can only really understand from that evolutionary history. For example, how the structure of an orchid relates to one of its pollinators and how those have co-evolved in time. But I think there are other questions or laws or answers that we can find by thinking about what is shared in common across the huge range of diversity of life, right? And I think one of the challenges of thinking about individual physiology or the specifics of individual species is that it often makes it very hard or it obscures the types of principles we might be looking for in trying to build new types of life or understand what life looks like in general. And so today I'll start by focusing on this question of what can we learn by looking across the vast diversity of life? And what are the right ways to look across the vast diversity of life? Are there ways to extract simple mechanisms to understand sort of universal regularities about what's going on for all organisms? And what's nice is that there are some of these answers starting to be observed. So for one example is if you plot the logarithmic mass of an organism, so this is just whole body mass, which would be the whole cell mass for single cell organisms, and all of the cells for multicellular organisms like you or me. If you plot that mass against the biomass production rate, and this is just mass production per individual per year, you see that across the huge range of diversity of life, you get a single relationship, and it's a power law. So you see that approximately you have that biomass production rate is equal to mass to the 3/4 power, right? And so this is sort of amazing for two reasons. One is that from a sort of physics perspective, we would say when you see a single relationship like this across so many orders of magnitude, it really hints at some sort of underlying law. And two, it says that this law is manifested through all of these detailed evolutionary processes I've been talking about, right? So at the largest scales of evolution, there seems to be a single relationship that all of life is conforming to. Now, I want to note that there's a huge range of variation around one of these curves, right? So one of these data points could be an order of magnitude away from the central curve, and that is where the detailed physiology and the diversity of species comes in. So this picture both tells us on average what's changing across huge changes in ranges of size, and still allows for the very detailed differences of physiology that we know to be true at the species level. And we get to see both in this picture. So There's then lots of work to do in uncovering what sorts of mechanisms lead to a law like this, and then lots of work to do about how do we take a law like this and use it to predict or implement detailed things in biology. If we have a detailed question or a detailed goal, how do we use a relationship like this? And that's really what I want to focus a large part of this talk on, is how do we apply these general laws to the specific problems we care about, either for cell physiology or bioengineering or understanding ecology. And so to do that, I'll take us through a set of sort of detailed stories about places where we can both understand mechanisms leading to these scaling relationships, and then the types of things that we can predict about biology from such scaling relationships. So to do that, let's zoom in a bit more on the small end of life. And what's interesting here is that now you see both these scaling relationships. Here we're plotting the volume of an organism against its metabolic rate. So this is how many watts it's using, which is just joules per time. And you can see that these different categories of organisms, whether it's single cell bacteria, single cell eukaryotic life, so unicellular eukaryotes, or multicellular eukaryotic life in green, you have a scaling relationship for each, which says that systematically across these organisms, there seems to be one set of mechanisms operating to tell you how much power they need as a function of size. But then we also see these scaling relationships shift as you grow across these taxonomic groups, which makes sense because we would say that if you have a mechanism that tells you something about how much power or energy you need at a particular size, that should be really strongly connected to the architecture and physical scales of an organism. And so as you go from the simple architecture of a bacteria to the slightly more complicated architecture of a unicellular eukaryote to the hierarchical architecture of multicellular eukaryotes, you should expect to see these radical shifts in the scaling relationships, which in fact we do see. Now, an interesting question becomes the one I asked just before this slide, which is how do we start to use this to make detailed biological predictions or understand what are trade-offs within physiology, or apply this in any number of ways we might be interested. And the simplest way is that we can start to build models of the things we most care about in physiology based on such scaling relationships. So one nice example would be to say that this total metabolism, which changes with the volume of an organism, is getting partitioned into growth plus maintenance, where this is this classic PERT idea where you need some amount of maintenance just to sustain the biomass even at zero growth. And then any extra energy you have above that goes to fueling new growth towards replication. And this simple budget we can write in a slightly more detailed way, where we say the total metabolic rate on the left-hand side, which is the scaling relationship with cell volume, equals the energy to create a unit of mass times the rate of mass creation, so that's just the biosynthetic term, plus the metabolic rate to support a unit of existing biomass times how much mass you actually have. And what's nice about this budget is we can then rearrange for dmdt, which is the growth rate, and solve the mass trajectory of organisms based on this scaling relationship and these two energetic parameters. And out of that, we get a fairly complicated form for the mass as a function of time, but it only depends on a few parameters. One being this ratio of basic energetic parameters, the initial size, and this metabolic scaling exponent. And then this initial allocation of energy to biosynthesis is connected to the energetic parameters in a complicated way that I won't describe here. But this does do a good job then of capturing both single cell and multicellular growth trajectories through a life cycle. So from the birth of an organism, which would be just after cell division in E. coli, to replication, or from an organism, say, hatching, growing through a life cycle and giving rise to new eggs. And in each of these cases, these trajectories match very well known energetics for these organisms. And so we can go back and forth between the growth rate of an organism and its energetic parameters, and we get a wide diversity of growth forms that range from being super exponential to turning over and actually having asymptotic sizes. And that all depends on small changes in the differences of these energetic parameters. And I see there's something in the chat. Yes, I'll come to what we can do for non-actively dividing cells at some point later. So I'll capture that question in a bit. Great. So this is nice. I think this connection back and forth with single cells or single individuals is great. But another nice thing that we can do is, since we know these mass trajectories, we can also solve for the generation time or the time it takes to have a reproductive event. And from that, derive population growth rates. And then what we see is that as you go across these major evolutionary transitions, you see these really different behaviors in how population growth changes with body size. In the bacteria, as cells get bigger, they're growing faster and faster. So the populations are doubling more quickly. And then in the unicellular eukaryotes, they grow more slowly as organisms get larger. And this is just due to the fact that these have slightly different metabolic scaling exponents between metabolic rate and cell volume, leading to these really different trends. And what's also nice is that beyond the simple trends, we also start to predict sort of hard limits to these two architectures. So we predict that at the small end of bacteria, you get this asymptotic behavior, which would predict a minimum cell size. And at the large end of unicellular eukaryotes, you also get this asymptotic behavior where growth rate goes to 0. And this would predict an upper bound on the largest possible unicellular eukaryotes. You can see that this occurs, this limit occurs sort of at the point where you stop seeing lots of single cell eukaryotes and you start seeing small multicellular eukaryotes. So we think this informs this evolutionary transition from single cellularity to multicellularity in the eukaryotes. And we think that this tells us something about the smallest possible bacterial cells. And what's interesting is that both of these asymptotes are actually being caused by the same phenomena. It's the point where maintenance metabolism takes over the entire metabolism, and cells can only sustain themselves. So this is that non-growing case, right? Everything the cell is doing is just for maintenance. There's no energy left for division. And this lower bound on bacteria agrees really nicely with new world record holders for smallest known bacteria coming out of Jill Banfield's lab. So this is our predicted smallest cell volume, and then this is the range of smallest cell sizes that they're seeing in this study from Jill Banfield's lab. And so this goes from about 1/3 of what we predict up to just about what we predict. And again, we're making an average prediction based on sort of average trends across all these organisms. So we're not, there's, if you sort of used min and max sort of bounding curves, then you easily get down to the sizes that were described in that study. So we think this tells us a lot about what limits the smallest, simplest cells, which is mostly that they're starting to be overrun by this demand for maintenance without the ability to harness enough energy to do lots of biosynthesis. I'm just going to take a peek at the chat, collect some questions. Yes, so I will come back to could this smallest cell be different in synthetic minimal cells? I think the answer is yes, if you can tune the maintenance values a little bit. And then we've done some work on on mixed populations and bacterial consortia. I won't talk about that here, but there are ways to start to use these ideas to build ecologies, which I'll touch on just a tiny bit at the end. Great. Okay, cool. So leaving this study, one of the things that we started to think about was, okay, so at the small end of bacteria and the large end of unicellular eukaryotes, they're both running into these challenges of maintenance metabolism, but what's happening sort of at the small end of eukaryotes and the large end of bacteria, right? So as the bacteria get bigger, everything seems better for them. They have more metabolic power. They're growing more and more quickly. Why don't they just keep getting larger and larger? And in that original paper, we said, well, it must be something to do with sort of basic biochemical processes. At some point, you're pushing these rates so fast that all the physiology and the basic biochemistry can't keep up with that. And so we assume that there must be some limitations in that system. And so what we wanted to do then is sort of go deeper into the physiology and not just look at energetics, but look at sort of all of the main macromolecular components and try and understand what's happening in those systems. Yes, so I'll talk a little bit about the JCVI cell a little later. So that's another good question. Great. So initially, we looked at some of this just phenomenologically, doing large compilations of data from the literature. And what we see, for example, is if you look at the total volume of DNA in a cell, and so this is really taking measurements for total mass of DNA in a cell and converting that into volume units to get a sense of how much physical space that's taking up in the cell. And so in all of these plots, I'll show a green line, which is just the one-to-one line between cell volume to show you sort of the upper bound on volume. And then you can compare the scaling of a macromolecule against that total cell volume. And so this would include multiple copies of a genome, small components of DNA, sort of everything. This is just extracting all of the DNA material and asking how much of it is in a cell. And what you see is that you get this very slow scaling with cell size. So the scaling is roughly a 1/5 power, which is very small. And so what it says is that as cells get bigger, as bacterial cells get bigger, they're sort of radically diluting out what the DNA concentration is for the whole cell. However, for the smallest cells, and you can see this little red point here for one of the Mycoplasma species, the DNA volume is roughly half the cell volume, right? So DNA is actually a huge fraction of the total cell volume. And if you extrapolated this curve down a bit, you would see that sort of just below the smallest observed cells, you run into a point where DNA would be predicted to take over the entire cell volume. Now we know that for most of these smallest genomes, every gene knockout is fatal. And so it may not be possible for other reasons to get much smaller than this, but even at this scale, the DNA space constraints are starting to be a real factor for these smallest cells. We can also look at the total expressed protein volume. So this is all of the expressed proteins in the cell, again, as a extracted mass converted to an average volume. And here we now see a much steeper scaling, but something that's still less than isometry, so still less than a power of 1. So again, proteins are diluting out as cells get larger. The exponent on this is more like 0.7. And again, at the smallest cell size, you would predict, again, roughly sort of half the cell volume that would be taken up by proteins. And if you go just a little bit smaller, again, you would run out of space for the expressed proteins, sort of extrapolating these curves. And so this is sort of interesting. One inference you can make from all of this is that the expressed proteins, the average copy number is going like the square root of cell volume. So it's not just that these proteins are growing because genomes are getting longer. It's actually because the number of times that each protein is expressed is increasing, but very slowly, like the square root of overall cell volume. Let me just check the chat again. Yes, lots of precognition in the, yeah, took me a second to digest that one. Great. So this is interesting. This is just sort of an empirical observation. But what we'd like to also start doing is building some theories about this physiology. And so one thing that we did in this paper was to say, can we build now a simple model for the ribosome requirement, right? And so what we did is we said, let's think about the ribosome dynamics over a cell cycle where the ribosomes have to make they have to replicate all the ribosomes, which means just replicating all the ribosomal proteins, and they have to build all of the rest of the proteins in the cell. And so the dynamics for the increase in the number of ribosomes is this production term of ribosomes, which is the number of ribosomes times how many base pairs a ribosome can process per second, divided by the length of all of the ribosomal transcripts put back to back, and then times gamma, which is just a fraction of the transcripts in the cell that are ribosomal opposed to all of the other protein transcripts. And then you have a decay rate because ribosomes break at some rate. So you get this specific decay of ribosomes. So this equation will just be exponential growth if you solve this differential equation, because it's just a constant times the number of ribosomes. And then you can have the coupled protein dynamics where now everything's the same, except you have 1 minus gamma. So all of the, all the ribosomes that aren't making ribosomal proteins are making all the rest of the proteins. You adjust by the average length of a protein rather than the ribosomal transcripts. And then you have this decay of proteins, right? And so these two equations can be solved together. You can optimize for gamma to make sure that these two pools replicate at the same time. And then you can solve for a required number of ribosomes that you need given a protein load. So given how many proteins the cell has, and given how fast you're growing mu, you can solve for the number of ribosomes. And so from everything I've told you so far, number of proteins changes with cell volume in a systematic way according to a scaling relationship, as does the growth rate. And then everything else in this equation is a constant. And so that allows us to predict how the number of ribosomes should change as a function of cell size. And then this is what we predict. So this is the curve for the predicted number of ribosomes. And then the points are the much messier data for how many ribosomes you have as a function of cell volume. And here I want to highlight that a lot of this noise is due to differences in experimental conditions, where of course the ribosomes are very sensitive to nutrient conditions. They're one of the main knobs that cells are using to sort of tune and respond to environments. And so, for example, this vertical line here is one study for E. coli across a range of growth media, which included having amino acids or not having amino acids in that growth media, and a whole bunch of other things, including even some dilute antibiotics. But anyway, on average, this model does a good job of telling us how many ribosomes it has. And most interestingly, it says that as cells get really big, the number of ribosomes you need sort of explodes, right? And you actually reach a point where you would need more ribosomes than can fit inside the cell. And that happens because the division timescale starts to approach the timescale for replicating a ribosome. So the time it takes a ribosome to make another ribosome with all those transcripts starts to be on the same order as the time it takes to divide the cell. That becomes impossible, and suddenly you need an infinite number of ribosomes. But this now gives us a hint as to what's starting to limit the largest bacteria, and it's this demand for replicative resources, namely ribosomes and all of the other RNA components that come along for the ride. I want to step back here for a second and just make a comment about how this interface from Terry Hua's lab. They describe this growth rate against sort of RNA to protein ratios, what should be the ratio of how much, sort of ribosomal material you have in the cell. And we recently wrote a paper talking about how if you include these degradation terms that we've described, you get the same answer for these growth laws at fast growth rates, but at slow growth rates, you predict a strong deviation away from these linear relationships, which seems to agree with E. coli and lots of other species at slow growth, because these degradation terms really start to matter. And that actually predicts a different optimal number of ribosomes. So this is a place where sort of a very detailed cell physiology can be described by these cross-species models by just taking sort of appropriate tuning for a particular species. Great. So coming back to this kind of broad picture of how the macromolecules are changing across cells of different size, what we see is that, I haven't told you about some of these macromolecular compositions, but they're in the paper. What you see is that the smallest bacteria are mostly DNA and protein inside an envelope. And then as cells become larger, you dilute out the DNA and the protein, and you become increasingly dominated by ribosomes and the associated RNA components. You can see the tRNA and mRNA curves along the same relationship. And eventually, that demand for replicative machinery becomes too much, and you get an upper bound on bacterial size. You also get a new lower bound here, because if we add all these components together, you can see that you run out of physical space at both the largest and smallest bacteria. So at the small end of bacteria, they're running into both space constraints and energetic constraints. And at the large end, they have no problem with energetics, but the space is becoming challenging to keep up with those energetics. And in between, you seem to have cells that have sort of lots of free space for water and so forth, which is E. coli, by the way, would be sort of at 10 to the minus 18 here. I know this is sort of a strange unit to put it in total meters cubed, but for a reference, E. coli would sort of be in the mid-range of this curve, sort of lots of extra free space. Yes, another bit of precognition, Petra. I'm going to come a little bit in just a few slides to sort of alternative takes on the ribosome. Great. So yeah, so the question then becomes, knowing this about what changes across bacterial space, across bacterial sizes, can we start to come up with some design constraints for bioengineering? And I really do mean, I don't mean intelligent design, I mean design from the perspective of people who are actually trying to build new types of cells. And so I think using the word design is appropriate in that sense. And so what are some of the design constraints that we can start to form from this? Well, what we really want to do is start to ask questions then about how much one could modify these scaling relationships, how much one could modify particular features at a given size, how much different origins of life or different evolutionary histories of life might change these relationships. And we really want to sort of start to define a very general but hard boundary for sort of all physiological possibilities in some sort of abstract space. And so we've been doing a little bit of this work where we started to ask questions about what changes for cells if you had a slightly different ribosome. So this could be if evolution discovered a different one, or if you engineer a radically different ribosome, what happens? And one nice example is if you just said, everything else about the cell is the same, except we vary how fast the ribosome is at processing a base pair. You could think about energy trade-offs here as well. So you could think about sort of speed energetics relationships. We've thought about that some as well. But here, just varying speed alone, you see that if you found a ribosome that was 10 times faster, you would get an extra order and a half of magnitude on the largest cell sizes. So bacteria could get much larger. And if you had a ribosome that was only 40 times slower, you wouldn't be able to encapsulate life at all, because you would always need more ribosomes than are available to the total cell volume. And so this is sort of an interesting way to start exploring this fairly broad space of tweaking different aspects of the biochemistry and asking what happens. One thing that one can also do then is ask how likely this is to be in the space of all things that we would find that do what the ribosome does. And I think the 10x faster is probably pretty unlikely. I think the ribosome we have is probably pretty close to optimal, and there's lots of studies that talk about that. One study that we talked about was to say, let's look at the energetic efficiency of a ribosome and ask, how good is it at performing this process of taking an unordered set of letters and writing them into a particular string? And the reason we frame it as this sort of problem is that then we can start to use all of this work that comes out of energetic costs of computation from the thermodynamic literature, which basically tells you if you define any abstract computation, you can bound the amount of energy that process uses by just knowing something about the change in entropy that algorithm is performing. And so if we say this is really just an entropic change from a pool of amino acids into strings, then what we can do is calculate sort of what the upper bound is for how much energy this process would take by any physical device writing any set of strings. And then we can compare that to what the ribosome actually does. And we find that the ribosome is only about 25 times less efficient than this ultimate physical bound. So the best that any device could do at writing strings in some way. And 25 may seem like fairly far away from the limit. This is actually an upper bound. the true energetic, the true calculation is likely to be much closer, because we really wanted to sort of upper bound this. So it's at worst 25 times less efficient than the physical limit. And as a reference point, computers are 100 million times worse. So life is doing, the ribosome is doing much better than even what we consider to be sort of highly engineered and designed computers at just writing strings, which is, I think, really interesting. So I think life is unlikely to find a better ribosome. We may have one that's pretty close to optimal. And so that's a nice thing to know in the space of biochemical possibilities. Another place, I'm going to skip this actually. I'll just say quickly that the maintenance metabolism, as it related to these relationships and predicts where these asymptotes are, is something that one can break down into all of the detailed processes of the cell. And in this paper, we did that. We sort of account for how much of cell metabolism, how much of maintenance metabolism is being used for protein repair, being lost to proton leakage, a whole variety of processes. And so I won't take you through that, but this is just to say that we can sort of do a detailed accounting of what these different repair processes look like. And you see that at the small end of cells, it's mostly protein repair and proton leakage that is accounting for maintenance metabolism. And then some of the ribosome components have sort of these very strange relationships for what fraction of maintenance metabolism they are. But at the large end of bacteria, they also explode, becoming sort of very substantial costs for the overall cell. I want to quickly mention some work that we've done with Sarah Walker's group, thinking about other types of functional scaling. So I've told you a lot so far about macromolecules, but one can also think about enzymes sorted into different functional categories. And here I'll just point you to this paper where we've looked at some broad functional categories, and we either see that you get sort of universal scaling across all these different categories, which says that for certain classes of function, you're not changing the ratio of those or the concentration of those relative to how everything else is changing. Whereas other classes really have big differences between how, say, the bacteria and the eukaryotes scale. And that tells you that there's some broad shift in physiology and function as you grow across that evolutionary transition. We're trying to understand what mechanisms are telling us why some of these categories should and shouldn't change across that evolutionary transition. And finally, I want to take this back to the beginning where I was saying that these many of this sort of detailed physiology or these scaling relationships is connected to environments. We know that evolution is situated in organisms adapting to particular environments. And so how do we start to make a link between, say, any selective pressure, which could be one that you set up in a lab, or one that happens naturally for long-term adaptation in environment, to these sorts of relationships? And so one way to do that is to start to ask, if you want to have these macromolecular scaling relationships, what do you need from an environment to make that possible? And so a very classic perspective on this is just to think about elemental ratios. So just what is the sort of whole cell stoichiometry as a function of different physiology or different macromolecular compositions. So we can take this set of relationships for each of these macromolecules, combine it with sort of average known stoichiometries for each of the macromolecules, and then just do a simple sum for each element over all the macromolecules as a function of cell size. And then from that, we can tell you sort of the required whole cell elemental ratio based on how large that cell is. And what we see looking at, say, nitrogen to phosphorus is that you get this really interesting non-monotonic function of cell size, where there's a maximum N to P in the mid cell sizes, and then you fall off for really large and really small cells. And I've shown here in black and gray, just the difference between gram-negative and gram-positive bacteria, just to show you how much those differences in the outer envelope matter for these stoichiometries. And then the dashed line is the Redfield ratio, which is this classic observation that if you look at nitrogen to phosphorus in the global ocean, you get roughly 16 to 1 across huge ranges of ocean environments. And so what I think is really neat about this is that we've gone bottom up from macromolecular scaling to sort of whole cell elemental ratios, and then explained why a wide range of cell sizes would give you something that looks like this globally observed Redfield ratio. So that could tell you a lot then about how if you had different macromolecules, if you pick different macromolecules, you could get different potentially global elemental ratios, or how different global abundances of elements might select for one macromolecule over another. And there's a really interesting feedback between those two. We think that could be a really interesting avenue for astrobiological biosignatures, which is what we mostly focus on in the paper that I'm showing here. And in general, this sort of work provides a foundation for building ecosystem models where you understand how organism physiology connects to an environment, how they come to a sort of a co-steady state for elemental abundances in cells and in an environment. And then you can sort of compare what sorts of cell N/P versus environment N/P ratios you'd get as a function of cell size, where you can see that on average, the largest cells would drive N/P ratios within the cell to be very different from that in the environment. The dashed line here is just the one-to-one line. And so the largest cells would sort of typically look the most different from their environment in terms of these elemental ratios, which again could be an interesting biosignature. It also could tell you something about different sorts of selective or adaptive pressures for picking different macromolecules, or how a choice of macromolecules will cause an environment to reach different steady states. And in general, sort of zooming all the way up to the top level of ecology, these relationships are very useful for predicting all sorts of things about macroecology. So we've used effectively these same sorts of models to predict the abundance of organisms you have on terrestrial landscapes for mammals as a function of body size, in good agreement with data. We've used this to predict the spatial patterning of lots of non-motile organisms, ranging from bacterial biofilms to forests to the location of termite mounds. We can sort of get this broad spectrum of spatial patterning ranging from random to totally hexagonally packed. And we can also predict long-term evolutionary trajectories between things like what types of resources you're targeting in terms of how patchy they are in your environment versus your body size. And here we're able to predict sort of the long-term trajectory of different categories of mammals. And so I think this is a really nice theory that can transition you back and forth between detailed questions about detailed physiology all the way up to sort of ecosystem level questions about competitive advantages and trade-offs and so forth. I just want to sort of mention that at the end for a bit of flavor. So from a more sort of detailed cell perspective, the parting thoughts I want to leave you with is that from these macroscopic laws, we can often infer simple mechanisms and understand some of the dominant trade-offs facing organisms at categorically different sizes. And we can also use this to answer a large variety of detailed questions about cell physiology, building new types of cells, and even asking questions about ecology. With that, I'd like to thank a long list of really fantastic collaborators. And I haven't included everyone on this list that's involved in this broader set of work. These are the people who are responsible with leading different aspects of the papers that I've shown today. And I would be happy to take more questions and come back to anything that I haven't answered and just engage in some discussion if people have that. So thank you very much for your time. And I will go to the chat to see what we have there.
Thank you very much. Yes, there are a couple more questions in chat.
Yeah, so the So it's interesting, there's a question about surface area to volume ratios and turning max size limit. They're really interesting and long-standing arguments about surface area to volume ratios. I think for lots of these limits, they're not the primary thing. And the reason for that is that in the smallest cells, you don't have a problem with, say, external gradients for diffusion of resources to the cell. And at those smallest scales, motility is incredibly expensive as a fraction of cell size because cells are living in such a viscous environment relative to their metabolic rate. So their motility is very expensive relative to total metabolism. And you're starting to run up into the level where diffusive forces really matter for motility and are actually killing you. Then as cells get much bigger, external diffusive gradients start to become really important for getting resources to the cell, but that happens as motility starts to become really inexpensive for cells. And so their ability to search the environment for resources becomes much greater, and that sort of external diffusive limit matters less. So that's one type of sort of place where people have thought a lot about surface area to volume ratios. And in this case, I think it's actually not the killer for larger cells because they can start to search their environment using motility. And so I think it's actually not the primary limit for what starts to be hard for bacteria as they get larger. And at the small end, I don't think it's much of a problem there. So yeah, thanks. Other questions? Anything else I missed in the chat? Yeah.
I have a question, Chris.
Yeah, thanks, Kate.
Would it be possible if you're trying to simulate or calculate the maximum and minimal size, would it be possible to kind of build a adaptive model that takes out different parts of metabolism to see how that affects it? That kind of goes back to the JCBI problem.
Yeah, absolutely. So, so that's something we've gotten very interested in, is trying to actually do the adaptive dynamics for some of this physiology. So say, taking all the constraints that we know, taking the starting point, and just asking simple questions about, okay, how do these organisms of different size compete against each other? Are they actually at evolutionary steady states? What's happening for the smallest organisms, and what are various trade-offs that could be made at those scales? Because there may be multiple sort of solutions. If the trade-off space is sort of large and complex, there may be multiple solutions at the small end for dealing with things, right? So if you can find a genome set or a gene set that's slightly smaller, that really makes your DNA volume a little bit smaller, that matters a lot, then suddenly you can express more proteins, you might be able to grow a little bit more quickly. And so there's all sorts of sort of interesting constraints there. And one could also think about pairing that with some of these whole cell or flux balance analysis for whole genomes to understand which sets of genes are possible and then what sort of downstream consequences that would have picking each of those sets. I think there's a really rich space of questions to ask there about different trade-offs and different things that might evolve. And if anyone's interested in that, please reach out, because we're thinking a lot about that recently. So yeah, thanks, Kate.
Thank you. I have many more questions that I'm going to bother you later about, but I don't have any right now. Does anyone want to unmute and ask a question, or we're done with chat? Okay, I think.
Hey, Kate.
Oh, Lynn.
I'll go ahead and say something. Hey, Chris, that was
Lynn,
a
nice to see you.
terrific talk. Lots of interesting stuff. Good to see you, but I am going to push back on the surface area to volume ratio, which I believe is the answer to all life's problems. And I think what you're doing is focusing on organisms that are actively out there swimming. And the obvious counterexamples are things like acetabularia, which is non-modal, but enormous single cell with a very long, central stalk. It's a unicellular alga. Neurons can be a meter in length. They're obviously not modal. And so I think your issue is not the swimming for the surface area to volume ratio, but it's gas diffusion. And I'm sure that there's been work published on that over the years. And if it's not gas, it's if you're heterotroph, it's still transport of other materials in and out of the cell. So, you know, I think here that is still very much key.
Yeah. So yeah, great point. So some interesting things there. So so one thing to note, I should have said this, is that everything I'm talking about here would be at maximum metabolic rate, maximum growth rate. And so there's a few interesting things that come into play. So some of the very large bacteria, which actually are larger than that upper bound that we described, are things like Thiomargarita, which is this sulfur reducing bacterium that tend to live in fairly energetically poor environments. And so therefore they're regulated to have these really low growth rates, right? And so their metabolism is already kind of super low. And so there's not much they could do to invest in motility, and they wouldn't get much return on investment there anyway. And if you tune those metabolic rates to be much lower, that same diffusive problem for external exchange also becomes less problematic because you're driving those rates at a smaller value. And the gas exchange would also sort of be aided by swimming as well, because you're swimming into these new environments without that external gradient. So I think there is a whole dimension there where you could rescale everything by the metabolic rates, and then talk about whether you need motility or not, and whether external diffusion is a problem or not. But yes, I think that there's a bunch of interesting things to talk about there. And then for neurons, you know, the interesting thing about what mammals are doing is, you know, we have all of this active transport and facilitated transport to deal with all sorts of problems, right? So a lot of those multicellular scaling relationships have been predicted by understanding what would be the optimal vascular network inside an organism to distribute resources, to actually pump resources to all of the individual cells. And that's where you get that sort of three-quarter power scaling. And that is in response to needing to actually do active transport to keep all of the cells running at a reasonable rate, given that diffusion would be absolutely impossible in something of our scale. So yes, some very interesting things, you know, subtleties to consider there about rate rescaling and when you start to invest in active transport. So yeah, thanks, Lynn.
Thank you. Thanks, Lynn, and thanks everyone who asked questions. And thank you so much, Chris.
Yeah, thank you, Kate, for having me. Thanks everyone for the great questions, and yeah, see you all somewhere sometime.
See you.
Yes,
Bye.
bye.